Imagine a 7-month-old infant, right?
Yeah.
Just lying there in the emergency room.
Oh, wow.
Yeah. And the baby is on his back in this pithed frog position.
Yeah.
Legs just splayed out, absolutely refusing to move.
Because it hurts too much.
Exactly. Every time a nurse even brushes against his skin, he just screams in pain. And you run a physical exam and find extensive bruising all over his tiny body.
Which is terrifying for a parent.
Completely. So you run every modern diagnostic test you can think of, only to discover that this infant is suffering from a disease famous for, killing 18th century pirates.
Scurvy.
It is, it's a profound shock to see that in a modern clinical setting.
Right.
And what makes it so fascinating and terrifying, really, is that this entire systemic collapse, the bleeding, the agonizing pain, it all stems from the absence of one single molecule.
And that is exactly the journey we are taking you on today. Welcome to our deep dive into the molecular foundations of human health.
We get a lot of ground to cover.
We do. We are pulling directly from the USMLE Step 1 lecture notes for Biochemistry and Medical Genetics. And our mission here is to take what is traditionally an incredibly dense textbook-heavy subject and synthesize it into a clear clinical framework.
Yeah, it's not just about memorization.
Exactly. So whether you are a medical student prepping for boards, a young professional in the biomedical field, or just an intensely curious self-taught learner trying to understand how your own body operates, we are going to explore how tiny, invisible molecular shifts dictate massive clinical outcomes.
Because, if you want to be effective in medicine or simply well-informed about human biology, memorizing pathways is just not enough.
It's just trivia at that point.
Right. You have to understand the why and the how. You have to see the direct causal link between a missing enzyme and a patient's actual suffering.
Okay, let's unpack this. To understand how the body breaks down, we have to start at the absolute foundation of how it's built. How is the information that makes you actually stored and expressed?
Which biologists call the central dogma of molecular biology.
Yeah, and I find it helps to visualize a highly secure tech company. Your DNA is the master source code. It is irreplaceable, and it is locked away in a highly secure server room, the nucleus of the cell.
Right, because you definitely don't want your master source code exposed to the volatile environment of the rest of the cell.
Exactly. So when the cell needs to build a piece of machinery, it doesn't move the DNA. Through a process called transcription, it compiles a temporary working copy of the code.
And that temporary copy is RNA.
Yep. The RNA is then securely transmitted out of the server room and into the main factory floor, which is the cytoplasm. And there, a process called translation reads that RNA code to assemble the physical hardware, the proteins.
So it goes DNA to RNA to protein.
You got it.
It's a brilliant system. But, if you spend any time studying biology, you learn that nature really despises absolute rules.
Always. There's always an exception.
Yeah. The central dogma implies a strict, one-way flow of information. But we have to look at exceptions like reverse transcription.
Wait, hold on. You're saying the temporary software can actually go back and rewrite the master source code? That feels like a massive security flaw.
It is a massive flaw, and certain invaders absolutely exploit it. Retroviruses are the classic example here.
Like HIV.
Exactly. A retrovirus carries its genetic payload not as DNA, but as RNA. When it breaches a host cell, it deploys a specific enzyme to reverse transcribe its own RNA into a DNA intermediate.
Wow.
And then it violently splices that DNA right into the host's master genome.
That is wild.
But it's not just a viral parlor trick either.
Sure.
Human cells actually use a limited form of reverse transcription themselves to amplify highly repetitive sequences in our own DNA.
So the source code is a living, adaptable document, essentially.
Very much so.
Now, before a cell can divide and create a new cell, it has to copy that massive genetic document flawlessly. This is the cell cycle.
Right, the phases of division.
Yeah. And for a eukaryotic cell, so human cells, we have interphase, which is the prep work that's broken into G1, then the S phase, where synthesis or DNA replication actually happens, and G2.
Plus a resting state called G0.
Right, can't forget G0. And finally, you have the M phase, or mitosis, which is the physical splitting of the cell.
And we should probably contrast that human complexity with something a bit simpler. Like if you look at a prokaryote, like a bacterium, its chromosome is just a simple, closed circular loop.
Just a ring of DNA.
Yeah, replication starts at one single origin point and just unzips in both directions until the forks meet.
But human DNA replication is vastly more complex.
So absolutely. It involves massive linear chromosomes and multiple origin points firing simultaneously. unzipping the DNA semi-conservatively.
Meaning, when the parental DNA unzips, each old half acts as a template for a new half. It's really elegant. But here's my question for you. If I'm a clinician standing in front of a patient, how does knowing these specific phases of the cell cycle actually help me treat them?
What's fascinating here is how we leverage this exact cycle to fight cancer. Because cancer, fundamentally, is just unregulated cell division.
It's the cycle gone rogue.
Exactly. A healthy cell cycle is regulated by strict checkpoints.
Uh,
The cell assesses itself like, is the DNA damaged? Do we have enough resources? If the answer is no, it halts.
it hits the brakes.
Right. But cancer cells mutate to ignore those checkpoints. They just keep dividing. So modern pharmacology uses drugs designed as essentially molecular wrenches to jam very specific gears in this cycle.
Break down how that works in practice. Like, if I have a patient on chemotherapy, what are these drugs actually doing at the cellular level?
Let's look at the S phase first, where the cell is desperately trying to synthesize new DNA. If we administer agents like methotrexate, 5-fluorouracil, or hydroxyurea, we are essentially starving the cell of the raw materials it needs to build DNA.
So the cancer cell tries to copy its code, runs out of supplies, and triggers its own death.
Exactly.
And what if the cancer cell manages to get past that S-phase blockade?
Then we target the later phases. For example, Blysin attacks during the G2 phase, which is that final gap before division. And if it makes it to the M phase, where the cell physically tries to pull itself into two new cells, we use drugs like paclitaxel, vincristine, and vinblastine.
I've heard of those. What do they do?
These drugs physically destroy the microscopic ropes, the microtubules that the cell uses to pull the chromosomes apart. So the cell is just paralyzed mid-division.
Wow, literally stuck.
Yeah. And finally, we have the real heavy hitters like cyclophosphamide and cisplatin. These are non-cell cycle specific.
Meaning they don't care what phase the cell is in?
Right. They cross-link the DNA, physically gluing it shut so it can't be read or copied at all, regardless of the phase.
It's just chemical warfare targeted at the machinery of replication. But, you mentioned something crucial there, starving the cell of raw materials during the S phase.
The building blocks.
Right. Before you can build a new DNA strand, you need the physical bricks. In biochemistry, those bricks are nucleotides, your purines and pyrimidines. So where do we actually get them?
Well, the foundation starts with a sugar called ribose 5-phosphate. This is generated by the hexose monophosphate shunt, which you can think of as a metabolic side street off the main glucose highway.
A side street, I like that.
Yeah. And this sugar gets activated by cellular energy to form a really critical molecule called PRPP. Once you have PRPP, the cell essentially has two options for building its nucleotides.
It can build them from scratch, or it can recycle. The from-scratch method is the de novo pathway, right? Which takes a lot of energy and mostly happens in the liver.
Direct.
But the recycling program is the salvage pathway. The cell takes leftover purine and pyrimidine bases from dying cells or degraded RNA and just reattaches them to PRPP to make fresh nucleotides.
And you might think recycling is just a nice bonus for the cell, like going green. But in certain tissues, the de novo pathway simply cannot operate fast enough to meet demand.
So the salvage pathway is basically essential for surviving.
Absolutely essential.
Which brings us to a truly devastating clinical scenario in the text. What happens when the cellular recycling program breaks down?
It results in Lesch-Nyhan disease. This is a severe genetic disorder caused by a deficiency or sometimes a total absence of a single enzyme in the purine salvage pathway.
HGPRT.
That's the one.
And if the recycling plant shuts down, the trash has to go somewhere, right?
Precisely. Because the body cannot salvage these purines, they accumulate and degrade into uric acid. So the patient develops severe hyperuricemia, which crystallizes in the joints, causing crippling gout.
Even in kids.
Yes. But the neurological impact is the true tragedy here.
Yeah, how does a nucleotide recycling issue destroy the brain?
It's all about the basal ganglia. That's the region of your brain that smooths out and coordinates fine motor movements. And it relies almost exclusively on the salvage pathway for its purines.
So it has naturally high HGPRT activity.
Right, and when you remove that enzyme, those specific brain cells basically fail. The clinical presentation is profound spastic cerebral palsy, severe intellectual disability, and a hallmark behavioral symptom that is just heartbreaking for families to manage. Compulsive self-mutilation. Patients will involuntarily bite their own lips and fingers.
God, it is a staggering reality check. One missing microscopic enzyme in a recycling pathway fundamentally alters a human being's behavior and physical existence.
It really is.
Now, zooming back out for a second, Synthesizing DNA, building proteins, running these recycling plants, this all requires an astronomical amount of energy.
Huge amounts.
How does the human machine actually fuel itself?
We extract energy from our environment in 4 distinct, highly regulated stages. Stage 1 is just digestion. Your gastrointestinal tract physically and chemically dismantles complex foods into monomeric building blocks.
So proteins into amino acids, carbohydrates into glucose, fats into fatty acids.
Exactly. Then stage 2 takes place inside the tissues. We take those various building blocks and funnel them down into one universal metabolic currency, acetyl-CoA.
Acetyl-CoA. And that enters stage 3, the citric acid cycle, or TCA cycle. Here, the acetyl-CoA is oxidized. But the genius of this cycle is that the energy isn't just burned off as useless heat.
No, that would be incredibly inefficient.
Right. The cycle strips high-energy electrons from the carbon structure and hands them off to specific carrier molecules, reducing NAD to NADH and FAD to FADH2.
I love how you describe this.
Yeah, I always picture NADH and FADH2 as like cellular batteries. They've just been fully charged by the TCA cycle, and they carry that charge to stage 4, oxidative phosphorylation, via the electron transport chain.
The powerhouse stage.
Right. They plug into the inner membrane of the mitochondria, discharge their electrons, and that electrical current powers a literal microscopic turbine called ATP synthase.
Which turns out ATP.
The pure energy your cells use to do work, while using the oxygen you breathe as the final trash can for those electrons, producing water as a byproduct.
That is the baseline for how we generate power. But survival also depends on how you manage stored power, particularly lipids or fats.
Okay, think of your body as having a very strict bank manager. When you eat a big meal, your blood sugar spikes and your pancreas releases insulin.
Deposit slip.
Exactly. Insulin tells the tissues, we are flush with cash, lock it up. Specifically, insulin suppresses an enzyme in your fat tissue called hormone-sensitive lipase, or HSL. It prevents any withdrawals from your fat reserves.
It keeps the fat locked away.
But imagine you are fasting or suddenly running a marathon, insulin drops. Stress hormones like epinephrine and cortisol flood the system. They act as emergency override codes. They activate HSL.
And once HSL is activated, it acts like molecular scissors on your stored fat. It hydrolyzes triglycerides, cleaving them into 2 parts glycerol and free fatty acid.
Then they go separate ways, right?
Right. The glycerol is shipped off to the liver, which uses it for gluconeogenesis.
So literally synthesizing brand new glucose to keep your brain fueled.
Exactly. Meanwhile, those free fatty acids bind to a carrier protein called serum albumin so they can travel safely through the blood to your muscles and other tissues, and there they are burned for raw energy through beta oxidation.
And this specific fat-mobilizing pathway is something we actively manipulate with a really common over-the-counter vitamin, isn't it?
Yes. Niacin, which is vitamin B3. If you prescribe niacin in massive doses way beyond what you'd need just for basic nutrition, it acts as a powerful anti-hyperlipidemic drug.
Wait, how does a basic vitamin lower cholesterol?
It works by directly inhibiting that exact enzyme we just talked about, hormone-sensitive lipase.
Whoa, wow.
Yeah. If you block HSL, the fat tissue stops releasing free fatty acids into the blood. The liver looks around, realizes it has fewer fatty acids arriving, and subsequently packages and secretes fewer VLDL particles.
And VLDL is the precursor to LDL.
Exactly. So by shutting down the fat vault at the source, high-dose niacin ultimately lowers the bad LDL cholesterol circulating in your bloodstream.
It is incredible how you can hack one pathway to fix a problem totally downstream. But here's the catch with that entire metabolic assembly line.
What's that?
Glycolysis, the TCA cycle, nucleotide synthesis, they do not run themselves. The enzymes doing the heavy lifting are essentially useless without specific molecular keys to turn them on.
Ah, yes. We call those keys coenzymes. And the vast majority of them are derived from the vitamins we absolutely must absorb from our diet.
Right.
The pathologies we see in vitamin deficiencies perfectly map to the specific metabolic pathways that shut down without them.
Let's look at the water-soluble vitamins first. Take biotin. It's the essential key for carboxylase enzymes that add carbon dioxide to molecules, like pyruvate carboxylase, which is critical for making new glucose.
Right. And a true dietary deficiency of biotin is exceedingly rare because our intestinal bacteria actually make it for us.
Very convenient.
But there's a fascinating clinical quirk here. You can induce a severe biotin deficiency if you consume massive quantities of raw egg whites.
Like a hardcore bodybuilder from the 80s.
Exactly. Raw egg whites contain a protein called avidin, which binds to biotin with incredible strength, basically trapping it in the gut so you cannot absorb it.
Crazy. Then there's thiamine, vitamin B1. This is the key for pyruvate dehydrogenase, and alpha-ketoglutarate dehydrogenase.
The gatekeepers.
Yeah, the exact gatekeepers that allow glucose derivatives to enter the TCA cycle. If you lack thiamine, you essentially cut the power grid to your highly active tissues like the brain and the heart.
And we see this most tragically in chronic alcoholism, as alcohol directly impairs thiamine absorption in the gut.
Right.
This manifests as Wernicke-Korsakoff syndrome, presenting with severe confusion, abnormal eye movements, and irreversible memory loss. Or it can manifest as wet beriberi, which is high-output cardiac failure.
We talked about niacin B3 as a drug earlier, but metabolically, it's the precursor to those cellular batteries we mentioned, NAD and NADP.
Right, synthesized from tryptophan.
Yep. If you don't get enough, you develop pellagra. Med students always memorize this via the 4Ds: diarrhea, dementia, dermatitis, and eventually death.
Let's look at folic acid and vitamin B12. These two are intimately linked, and both are completely non-negotiable for DNA synthesis.
So if you lack them, cell division halts.
Exactly. Without them, cells try to divide but fail, leading to megaloblastic anemia, where you find giant, immature, and basically useless red blood cells in the bone marrow.
But B12 has an added layer of danger, doesn't it? Like if a patient is deficient in folate, they just get the anemia. But if they're deficient in B12, they also suffer severe, irreversible neurological damage. Why the difference?
That's because B12 is also the required cofactor for a totally different enzyme called methylmalonyl-CoA mutase.
Okay.
And that enzyme is vital for metabolizing odd-chain fatty acids. Without B12, abnormal toxic fatty acids build up and are mistakenly incorporated into the myelin sheaths that insulate your nerves.
Oh, man.
Yeah, the nerves literally short-circuit, causing severe peripheral neuropathy and cognitive decline.
Here's where it gets really interesting, tying back to the story we opened with. The 7-month-old infant screaming in the ER. Why did that baby have scurvy?
Well, scurvy is a severe deficiency of vitamin C, or ascorbate. Vitamin C is the required cofactor for prolyl and lysyl hydroxylases.
Big words, but what do they do?
These enzymes hydroxylate specific amino acids in collagen, which allows the collagen fibers to cross-link and form a really strong triple helix structure.
So without vitamin C, your body just builds defective, weak collagen. Your blood vessels lose their structural integrity and just dissolve, causing the extensive bruising and internal bleeding.
It falls apart at the seams.
Yeah. And in the case of this modern infant, the mother was exclusively bottle feeding him. She was so terrified of bacterial contamination that she was boiling the commercial formula for extended periods to sterilize it. And ascorbate is highly heat sensitive. By boiling it, she completely destroyed the vitamin C, inadvertently inducing scurvy.
It is a perfect, albeit tragic, example of why exogenous cofactors are essential. Just to briefly complete the picture, we also rely on lipid-soluble vitamins.
A, D, E, and K.
Right. Vitamin D acts almost like a hormone to regulate calcium and bone density. Vitamin A maintains healthy epithelial tissues and is crucial for retinal function and vision.
What about K?
Vitamin K is the necessary cofactor for carboxylating glutamic acid residues in your clotting factors. Without it, your blood cannot coagulate. And finally, vitamin E acts as a biological antioxidant, protecting your delicate cell membranes from oxidative stress.
Okay, so we've established how the DNA source code works, how the cell fuels itself to read it, and the vitamin keys required to run the machinery. But what happens when the master source code itself contains a typo?
This is the realm of medical genetics. We won't dwell on the basic definitions of alleles or genotypes since our listeners know the basics, but we really need to focus on how the DNA sequence is actually altered.
The mutations.
Right. We categorize these mutations by their impact on the final protein.
Okay, so a nonsense mutation is devastating. It accidentally creates a premature stop signal. The translation machinery is building the protein, hits the mutation, and just quits halfway through, leaving a truncated, useless fragment.
Very bad news.
And a frameshift mutation happens if you insert or delete a nucleotide, but not in a multiple of 3. Because the DNA code is read in 3-letter words, adding 1 letter shifts the entire reading frame down the line. It turns the rest of the protein into absolute gibberish.
And then you have missense mutations, which simply swap out one single amino acid for another. The classic clinical example of this is sickle cell disease.
How does one single amino acid swap cause such a systemic, painful disease?
It is just a simple missense mutation where valine replaces glutamic acid in the beta-globin polypeptide of hemoglobin.
Just one swap.
Just one. Glutamic acid loves water, valine hates it. Under low oxygen conditions, that single hydrophobic valine molecule causes the entire hemoglobin protein to polymerize.
It changes the shape of the cell.
Right. It physically warps the flexible red blood cell into a rigid sickle shape. Those rigid cells jam up in the capillaries, causing immense pain and organ damage.
If we connect this to the bigger picture, how does a clinician actually track these dangerous typos through a family tree?
We use pedigrees. The first patient identified with the disease in a family is called the proband. By mapping the family's phenotype, we determine recurrence risk.
Give me an example.
Let's say we're looking at an autosomal recessive disease like cystic fibrosis. If both parents are healthy but carry the hidden recessive trait, meaning they are both heterozygous, there is exactly a 25% chance with every single pregnancy that the child will inherit both mutated copies and manifest the disease.
The dice roll fresh every single time. So if we know a family is at risk, how do we actually find the typo in the lab? Are we searching the entire 3 billion letter human genome for one wrong letter? Or do we just look for the neighborhood the typo lives in?
That perfectly describes the difference between direct and indirect diagnosis. Direct diagnosis is used when you know exactly what the mutation is and exactly where it lives. You test specifically for it.
Give us an example of how you directly hunt down a known mutation.
One method is using allele-specific oligonucleotide probes, or ASO probes. Think of these as customized molecular magnets.
Okay, molecular magnets.
The text uses hemochromatosis and iron overload disorder as the example. The most common mutation is C2A2Y, which is a substitution of a G nucleotide for an A.
Got it.
The lab synthesizes two short radioactive DNA probes. One is designed to magnetize perfectly to the normal G sequence. The other magnetizes perfectly to the mutated A sequence.
So you wash the patient's DNA with these probes.
Exactly. And whichever probe sticks and lights up on the scan tells you exactly which alleles the patient possesses.
Another brilliant direct method uses restriction fragment length polymorphisms, or RFLPs. I love this concept. Restriction enzymes are basically molecular scissors that patrol DNA, looking for very specific sequences to snip.
Right.
Sometimes a disease mutation accidentally destroys a sequence the scissors normally cut, or creates a new one.
Exactly. In sickle cell disease, the mutation accidentally destroys a cut site for an enzyme called MTII. Gaucher disease, which is a lipid storage disorder, is another great example. A specific mutation there creates a brand new cut site for an enzyme called HVAI.
And you can visualize this, right? If you take that segment of the patient's DNA and expose it to the HAI scissors, a normal gene remains intact.
Right, no cut.
But if they have the Gaucher mutation, the scissors recognize the new site and chop that segment into two smaller pieces. When you run that DNA through an electrical gel, smaller pieces travel much faster than large intact pieces.
So you can literally look at the gel scan and see if the DNA got chopped up, instantly confirming the mutation.
But what if you don't know the exact mutation? What if a family has a rare, uncharacterized bug in their source code?
That is when we use indirect diagnosis.
Reading the shadows on the wall.
That's a good way to put it. The text details a case of long-chain acyl-CoA dehydrogenase, or LCAD deficiency. A couple lost a child to it, but standard direct tests show negative for known mutations.
So what do they do?
The lab uses indirect markers, basically harmless, random variations in the DNA that happen to sit right next door to the LCAD gene on the chromosome. By testing the parents, the healthy sibling, and the current fetus, they can track which specific chromosomal neighborhood the affected child inherited.
And see if the new fetus inherited that same dangerous neighborhood without ever actually knowing the exact typo.
Exactly.
Everything we just covered is looking at the master DNA code. But we can also look at the temporary RNA software, right? We use southern blots to look at DNA, but northern blots are used to extract and analyze RNA from a tissue.
Which is crucial, because a northern blot tells you not just if the gene exists, but if the cell is actively transcribing it.
Right.
For example, in Fragile X syndrome, which is the leading cause of inherited intellectual disability, the mutated FMR1 gene is normally highly expressed in the brain and testes. A northern blot confirms if the tissue is actually producing that massive RNA transcript.
And we are taking this to an entirely new level with microarrays. Instead of running a gel to look at one single RNA transcript, A microarray puts microscopic probes for hundreds or thousands of genes onto a single glass chip.
That's unbelievable tech.
You can take a biopsy of a woman's breast cancer tumor, wash its RNA over the chip, and the chip will literally light up to show you exactly which hundreds of genes the tumor has turned on and which it has turned off.
It really is the absolute pinnacle of applying the central dogma to clinical practice. You aren't just looking at the static blueprint anymore. You are watching the construction site operate in real time.
Allowing you to tailor a highly specific chemotherapy regimen to that exact tumor's profile.
Exactly.
It is staggering how far we've come. Okay, we have covered a massive amount of ground today, from the flow of genetic information to cellular fuel to finding the errors in the code. As promised, I want to leave you with a quick review exercise to solidify this.
Little pub quiz.
Yeah. Think back to our discussion on nucleotide recycling.
Sure.
If you are examining a pediatric patient presenting with severe gout, spastic cerebral palsy, and a tragic history of compulsive self-mutilation, what specific nucleotide pathway is defective, and what is the exact missing enzyme?
Take a second to think about it.
Right. Well, if you visualized the purine salvage pathway and identified the missing enzyme as HGPRT, you nailed it. That is the devastating cascade of Lesch-Nyhan disease. So, having covered all this, what does this all mean for the future of medicine?
It means we are fundamentally shifting from a static view of biology to a dynamic one. Genetic diagnosis is evolving incredibly rapidly.
Yeah.
We are moving beyond just finding the one typo in a single gene disorder like sickle cell. With tools like microarrays, we are now interpreting vast systemic gene expression profiles.
The big picture.
Right. As we enter the true era of personalized medicine, the ultimate question isn't just, what does your static DNA code say? Perch. The real question is, how is your specific cellular environment influenced by everything from the vitamins in your diet to your stress hormones, choosing to read and execute that source code today?
The source code is just the beginning. The real story is what happens on the factory floor. Keep questioning the mechanisms, keep zooming in on the why, and keep learning. Catch you on the next deep dive.